Good morning, Anir. Anir, now you can unmute and you can speak also. There is no problem.
Okay. Good morning. Good morning. Can you hear me? Okay.
வந்துருச்சு போட்டு விட்டீங்களா நவிதா யாரும் அந்த மீட்டிங்குள்ள எல்லாரும் வாங்க பாத்துக்கிட்டு தான் சொல்றேன் கூப்பிட்டேன். Good morning, Dr. Sugandha Mantal.
Good morning, Sundar.
Yeah, how are you?
Fine.
Okay. I think you can just we can check whether the sharing is there any problem.
Okay.
Just share your front page.
Can you see?
Yeah, it's coming. Yeah, I think there is no issues on it. It's fine.
Okay.
Yeah, fine. So 10 more minutes are there. I think we can start after that, sir.
Yeah, okay.
So very good morning to all. So on behalf of the Department of Chemistry, I welcome you all to the morning day 2, session 3. Today resource person is Dr. Sugandha Mantal from IIT Kharagpur. He is going to deliver the session 3 lecture. So may I now request Dr. T. Suresh, Assistant Professor, Department of Chemistry, Bharatiyar University, to introduce the speaker. So thank you.
Yeah. Good morning to all. Hope you people enjoyed yesterday 2 sessions. Okay, yeah. Today I'm happy to introduce our today's speaker, Dr. Mendal. He is currently working as an additional professor in IIT Kharagpur. About his career, he has done his PhD in IIT Kanpur. It is well-known, reputed institute in India. After his doctoral doctoral degree in IIT, he visited a couple of countries like Spain and South Korea for his postdoctoral research. He has received a DST Inspire Faculty Award in 2012. Also, he has earned CSR, SRF, and JRF fellowship during his doctoral studies. He has published his research findings in well-known reputed journals like JACS, Inorganic Chemistry, NJC, and European Journal of Chemistry. Apart from this, he has published one paper in Nature Chemistry. Okay, its impact factor is 21.75. Okay, this shows his excellent contribution in his field. He has handled 3 projects in SERB, CSIR, and ISRD SRC. Apart from this, he has visited Sweden country for his research collaboration. Okay, totally he is today he is going to deliver the lecture on water oxidation catalysis by coordination complexes. Okay, on behalf of FCS 2020 team, I am warm welcoming Dr. Mental. Okay, now I am handed over session to Dr. Mendal. Okay, Doctor,
Yeah.
please
So
proceed. Okay.
yeah, let me share first my screen. So it is visible.
Yeah.
Yes, doctor. Yeah.
Okay. First of all, thank you, Dr. Suresh, for your kind introduction. And it's my pleasure to be a speaker in this webinar. I especially thank Dr. Sundaravel and the other organizing members to invite me and giving me an opportunity to present some of my research work. So I hope in audience there will be many undergraduate students. So I will try to explain the chemistry in a very simple manner. So at least I will try, but I don't know how much I will be successful. So I will be mainly talking about coordination chemistry. So this particular branch area of chemistry that I liked very much when I was an undergraduate student. And as you know that this particular field was first introduced by a German chemist, Alfred Werner, and for that he was awarded a Nobel Prize in 1913. And since then it has been more than 100 years, and since then this particular area of research has evolved enormously, particularly in the field of catalysis. So in my presentation today, I will be talking about mainly one particular reaction, that is the oxidation of water by some coordination compounds. So obviously the inspiration of doing this such reaction comes from the nature. So as you know, nature is always being a role model for many of our many researchers. So as you know that water oxidation, it happens during photosynthesis by the green plants. And the overall photosystem can be divided into 2 parts. One is photosystem 2 and another one is photosystem 1. And this particular reaction, that is the oxidation of water to oxygen, it happens in photosystem 2. So here I am showing the crystal structure of photosystem 2. As you can see from this figure, that it's a very complicated molecule. It has a very complicated structure. But inside this protein cavity, you have one small inorganic component which we called as an oxygen evolving complex. And this oxygen evolving complex actually catalyze the water oxidation. And this is the crystal structure of this oxygen evolving complex. As in yesterday also, Dr. Shankar also showing this particular structure. So I hope you will be familiar of that. So this particular complex has a couple of manganese ions, and also you have one calcium ion. And when you flash light, this particular oxygen evolving complex is passed through various intermediates, which is called as a Koch cycle. It goes from S0 to S1 to S2 and S3. And when it reaches at S4 state, it acquires a lot of oxidizing power. Then it can promote this particular reaction, and it can oxidize water to make oxygen. So our goal in our research to develop a small molecular system that can mimic, obviously it is not the structural mimic, we want to do that functional mimic of photosystem too. So if you look at the literature, so there are two completely different approaches has been adopted to develop the catalyst to promote these particular reactions. So one approach is use of metal oxides, metal oxide nanomaterials as a catalyst to do the oxidation of water. And this particular approach is quite old. It began in 1902. And there are another approach where we can use a discrete molecule, a discrete coordination compounds, which we can use as a catalyst. And this approach is comparatively newer. So Professor T.J. Mayer, he used this particular ruthenium dimer, which also called as a blue dimer. And when he has used a steric ammonium nitrate in an acidic medium, it was shown that first time that this particular complex can evolve oxygen by oxidizing water. So both the approaches has some advantage, also some disadvantage. The main advantage of this molecular approach that it is very easier to investigate the mechanistic aspects. So once we can understand how this reaction is going on, then actually we can tune the ligand system and we can tune the catalytic reactivity. But the main disadvantage of this particular approach is that these organic ligands, they are prone to be oxidized under very oxidizing condition, and by that way the catalyst gets deactivated. So as my title says, so I will be mainly talking about this molecular approach. I won't talk about this oxide approach. So this is the outline of my presentation. So first I will give an introduction, background knowledge about this particular field, and then I will show some of the research outcome from my IIT Kharagpur lab, as well as I will also share some of my work which I did during my postdoctoral study. And then I will conclude with some summary along with formal acknowledgement. So my student, when they give synthesized a compound where you have a water ligation, so I always get very much excited, because that type of compound I can use, it shows very interesting properties, and I can use for various purpose, and and I can use it in for catalysis also. Like in this slide, what I am showing here, so when you have a water ligated metal compound, so you know that water in free state, it has a pKw around 14, but when it gets coordinated with the metal center, its pK value reduces enormously. So what does it mean? That lower pK means this hydrogen atom becomes more acidic in nature. So then what you can do, just by using a acid-base reaction, you can generate a hydroxide, metal coordinated hydroxide nucleophile. And such type of species can do hydrolysis of many important biomolecules. Actually, nature use this type of hydroxide, metal coordinated hydroxide to hydrolyze various biomolecules which mimic the metallohydrolases. In another way, I can also utilize the water for in PCT reactions. PCT reaction means it's a proton couple electron transfer reactions. So I can involve the water in proton couple electron transfer reaction, and I can generate a high valent metal oxo species. So in yesterday's two lectures, we have learned more about that such type of species. So this type of species has a very much oxidizing power. So it can oxidize, we can use for various oxidative transformation reaction, like we can oxidize, do the sulfoxidation reaction. We can functionalize the CH bond to make alcohol type of value product. We can do some epoxidation reactions. So those type of reactions, our yesterday speaker has already been explained to you. But today what I am going to show you, that by using such type of metal oxo species, we can do a very challenging reaction, that is the oxidation of water to make oxygen. So my objective will be try to understand how these particular reactions we can do it, and what are the things we need to understand if we want to develop a artificial photosystem device that can do efficiently this oxidation of water to make oxygen. Now questions comes, why suddenly we become interested for these particular reactions? So the reason behind it, if you want to make an energy economy based on water and sunlight, so then we need to understand these particular reactions very well. Like in this scheme, what I am showing, so with sunlight, I can split water to make hydrogen and oxygen. So by that way, the electron, I can store the electron from the water, I can store into the hydrogen. So then hydrogen I can use as a fuel. So when you combine this hydrogen and oxygen in a fuel cell, we can produce electricity. So from this scheme, I can actually utilize solar energy in the form of a electrical energy. But to split this particular water to hydrogen and oxygen, we have to develop another device. We have to fabricate another device. So this I named as the artificial photosystem device. So this particular device actually contains three major parts. One is photosensitizer. This PS is photosensitizer that actually absorb or harvest solar energy. So in nature, that part is done by the chlorophyll. And in our artificial system, we can use various types of dyes or semiconductors. So when it absorbs light, a charge separation takes place. So holes generates, and that hole can then oxidize water in the presence of a water oxidation catalyst to make oxygen, along with 4 electrons and 4 protons. And these 4 electrons and 4 protons you can then recombine in a cathode reaction in the presence of a proton reduction catalyst to make hydrogen. So that means from the electrochemical point of view, this splitting of water can be divided into two half cell reactions. One is anode reaction, where we oxidize water to make oxygen along with protons and electrons. And these protons and electrons which you get from the anode reactions, that can be combined in a cathode reaction to make hydrogen. But out of these two reactions, the bottleneck is the anode reactions. This is the most challenging part, because it has a it is energetically uphill, as well as it has a very high potential. Its potential is around 1.23 volt at pH 0. So that means if you dream to drive our car instead of a petrol, but to use with water, so that means we have to install this fuel cell as well as the artificial photosystem device in our car. But we have to develop a very efficient catalyst for this water oxidation part, because this is the most challenging part in this energetic scheme. And this energy scheme is very attractive, because here we don't produce any harmful byproduct. We only produce water, and by that way we can replace our carbon-based fuel energy economy with this water sunlight energy economy. And by this way, we can actually checkmate the production of carbon dioxide, which you know that this is the responsible for the global warming things. Now to do that, let me understand these particular reactions at molecular level. So what are the things, how we can activate the water molecule so that we can produce an oxygen along with a proton and electron? So what are the steps we need to understand? So from the stoichiometry, as you can see, to make one molecule of oxygen, you need two water molecules. And then from each water molecule, first we have to think of how we can remove these protons and electrons from this water molecule. So once we can remove the proton and electron, then we can make a naked type of oxygen atoms. And then once we make the naked kind of oxygen atoms from the two water molecule, our second step would be to make an oxygen-oxygen bond to make oxygen molecule. So that means this particular reaction, there are two steps. So we have to activate our water molecule somehow so that we can remove its proton and electrons. And once we remove its proton and electrons, then second step would be a oxygen-oxygen bond formation. And I remind you that this oxygen-oxygen bond formation step is very kinetically slow process. As you know that this particular reaction, the delta G value is positive, which means it's not at all a spontaneous reaction, and it's a very energetically uphill process. So it has some thermodynamic barrier, as well as the oxygen or oxygen bond formation is very slow. That means it also has some kinetic barrier. So that makes this particular reaction even more challenging. So to develop, we have to develop a efficient catalyst so that, and we have to understand this particular reaction at a molecular level so that we can develop an efficient catalyst that can promote this water oxidation in a very efficient manner. So here I am showing how a PCET reaction, a proton coupled electron transfer reaction, can be very useful to activate water molecules. So in this scheme, what I am showing, so see, this is, suppose I have a aqua ligated ruthenium complex, then I can undergo this particular complex, a couple of electron transfer, and along with a proton coupled electron transfer, and make an high valent ruthenium oxo species. So if you look at when I'm going from left hand side to right hand side, where you have involved some sorts of proton coupled electron transfer reaction, by that way I am actually removing the hydrogen atoms from the water molecule. So once we have removed the hydrogen atoms from the water molecule, so then our next step, we have to think of how we can make the oxygen-oxygen bond. So here proton coupled electron transfer mechanism is very helpful to activate water so that we can remove its protons and electrons. So here I am showing one example how a PCET reaction can be beneficial from energetic point of view so that we can remove its proton and electron very easily. So let us take this particular complex where you have a ruthenium and this type of ligand, and you have a bipyridine, and you have one chloride ligation. But here you don't have any water ligation. So if you look at its electrochemistry, it shows two oxidative response. So first response we are getting from the ruthenium 2 to ruthenium 3, and second response we are getting from the ruthenium 3 to ruthenium 4. And if you look at this Pourbaix diagram, so both the redox couple, they are independent of pH, which means here only electron transfer is going on. We are not removing any protons. As this molecule does not have any water coordination, so we don't expect also that there will be some proton coupled electron transfer. Now, if you look at the relative potential values, the 3-2 potential is coming around at 0.1 volt. So when you apply a 0.1 volt, we remove one electron from this particular species, and it generates a ruthenium 3 species. Now, when you want to remove the second electron, you see I have to apply a very large potential. I have to apply all the way to 1.1 volt potential. And the relative gap between these two redox couple is around 1 volt. That means it requires a lot of energy when we want to remove its second electron. So because when it involves only electron transfer, so what is happening, you are developing an additional charge on the oxidized species. Now once you develop an additional charge, now to remove the second electron becomes very much difficult, and you have to cross a larger energy barrier to remove the second electron to form a ruthenium 4. But if you look at this particular complex, now I am taking this particular complex where I have a ruthenium, a terpyridine, and bipyridyl. Now I have an water ligation. Now, if you look at its electrochemistry, this red line represents the electrochemical behavior of this particular complex. So it's 3,2 and 4,3 redox couple. Now they are dependent on pH. So which means now here I am not only removing an electron, I am also removing an proton. So it's a proton coupled electron transfer reaction is going on. And if you look at the relative gap between these two redox couple, it's now around 0.1 volt. So I have reduced the energy barrier around 10 times. Okay. So why this is because when we have a proton coupled electron transfer, you see when you have a proton coupled electron transfer, then we don't develop an additional charge in our oxidized species. And when you don't develop an additional charge, that actually minimize the energy. And then we can remove the second electron as a comparatively lower potential values. So like that way, PCET is very helpful to activate the water so that we can remove its electron and proton from water molecule. Now, once we have removed the protons and electrons from water, now we have to think how we can make the oxygen-oxygen bond. So if you look at the literature, there are two possible pathways where how we can make an oxygen-oxygen bond. One pathway is water nucleophilic attack. We call this an WNA. And another one is I2M mechanism. We call this an interaction between two metal oxo units. Now, in WNA mechanism, if your metal centers acquires a very high oxidation state, then actually it drags lot of electron density from this oxo moiety. And then this oxo moiety becomes electrophilic in nature. So once it becomes electrophilic in nature, then we can expect a nucleophilic attack from water molecule, and it can then make a hydroperoxide type of intermediates, which then further undergoes oxidation to make oxygen molecule. And in I2M mechanism, so sometimes from the DFT calculation, we found that so this metal oxo unit sometimes possess a radical-like character on the oxygen atom, on this oxo moiety. So it is rather a metal oxyl type of intermediate. Now, once we have a radical character, so then two metal oxyl unit, they can combine bimolecularly, and then they can form a metal-metal peroxo type of intermediate. And then this peroxo intermediate undergoes further oxidation to form an oxygen molecule. So beside these two mechanisms, another pathway can be found, which I named as a I2MHC mechanism. Now, if you follow the literature of this particular area of research, here we use ferric ammonium nitrate as a sacrificial oxidant to oxidize our metal complex from lower valence state to higher valence state. Now, when we dissolve this ferric ammonium nitrate in water, so our reaction medium in this case is water, because we are doing the water oxidation. So when you dissolve this particular ferric ammonium nitrate in water, so one of the nitrate group that is actually hydrolyzed by water, and then you have a cerium 4 hydroxide type of intermediates. Now, from the DFT calculation, it was found that this cerium possesses a lot of Mulliken spin density, as well as this oxo also possesses a lot of Mulliken spin density, which implies actually the proper electronic structure of this species is not an cerium 4 hydroxide, rather it is a cerium 3 and hydroxyl radical. So once you have a radical character on hydroxyl, and if your metal oxo unit also possesses the radical character, then we can expect a heterometallic radical coupling can take place to form a oxygen-oxygen bond, and then it can form this type of intermediates, which then further undergoes oxidation to make oxygen molecule. Now, this particular type of mechanism, we can say there is some resemblance with natural photosystem too, because you know that in natural photosystem, the oxygen evolving complex, it not only possesses the manganese ion, it also has some calcium ion. So although it is not very clear to date what is the exact role of calcium, there is a lot of doubt of that, it's not very clear, but some researchers believe that that water molecule or hydroxide coordinated at the calcium center that actually involve in oxygen-oxygen bond formation with the high valent metal manganese oxo species at S4 state in Koch cycle. So with this background, let me now talk about my research, which I am doing at IIT Kharagpur. So we have developed a few mononuclear ruthenium complex. So these two complex we say is a first generation of catalyst in our lab. So we took a pentadentate coordinating ligand and we make a ruthenium aqua complex. So this ligand and so this complex, this is with N4-PY ligand. This is already reported in literature. We named it as 1 aqua complex, and then we designed a new pentadentate ligands in our lab. And with that ligand we make a ruthenium aqua complex, and we named it as a 2 aqua complex. So both the complexes have a similar type of coordination environment. So here you have a 1 tertiary nitrogen, and you have a 4 pyridyl nitrogen donor groups. But here you have a tertiary nitrogen, 2-pyridyl and 1 bipyridyl donor groups. And we studied their comparative reactivity towards water oxidation. So this is the synthetic procedure. So first we synthesize this chloro precursor, and then this chloro like ligand we can easily replaced by using a silver salt in presence of water to get our aqua ligated metal complex. And as this is a ruthenium 2 state, so it's a low spin D6, so we can characterize it's a diamagnetic in nature, so we can characterize easily by 1H NMR spectrum. It gives a very beautiful NMR spectrum between 0 to 10 ppm. With similar approach, so we also synthesized our second ruthenium aqua complex, and this also characterized by 1H NMR spectrum, and also this was also characterized by single crystal X-ray crystallography. Now, whether these two catalyst or complex is able to do the oxidation of water or not, for that we perform this electrochemical behavior, and this is the tool we use enormously in this area of research. So electrochemistry is a major part in our research in order to understand what are the how many oxidizing species we can generate, and what are the and which species is actually responsible or active species for triggering the oxidation of water. Now, if you look at that electrochemistry of this particular complex, now this red line is a blank one, and this black line is the electrochemical behavior of this particular complex. Now, you see that when we reach an 1.5 volt, it's producing a catalytic current, a huge catalytic current. So this catalytic current is actually implies that this complex is triggering the oxidation of water when we reach at potential of 1.5 volt. So we have performed a Pourbaix diagram. As you can see that this complex also shows a couple of proton couple electron transfer reactions, and then we ultimately generate a formal ruthenium 6 oxo species. Okay, so this is the comparative electrochemical diagram of these two complexes. The blue line represents for this N4PY complex, and the black line is for this one. From the catalytic current density, you can easily make out that 2 aqua complex is performing much better compared to 1 aqua. So this is the oxygen evolution profile. This is the oxygen evolution profile for this two complex. So we used steric ammonium nitrate as our sacrificial oxidant, and with the presence of catalyst, we monitor the amount of oxygen it evolved with time. So as you can see that this black trace, which is for the 2 aqua complex, is showing much better activity compared to our this 1 aqua complex, which actually corroborate very well with our electrochemical results. What happened? So further, in order to understand how this particular complex is promoting the oxidation of water, so we performed its kinetic analysis. So we perform the kinetic analysis by varying the complex concentration as well as by varying the steric ammonium nitrate concentration. So when we varied the complex concentration, we see that rate of oxygen evolution is dependent linearly with the complex concentrations, which indicates that the reaction is first order with respect to complex concentration. That means that only one molecule of water is actually performing the oxidation of water. It's only one molecule of complex is actually performing the oxidation of water. But when we varied the cerium concentration, we see it is independent, the rate is independent with the concentration of cerium. So that means the reaction is zero order with respect to cerium concentration. So we can write the rate loss, it is a first order with respect to our complex concentration, and zero order with respect to cerium 4 concentration. So zero order with respect to cerium 4 concentration means that at rate determining step, the cerium does not participate in rate determining step. So from this kinetic analysis, as it is a first order with respect to catalyst concentration, we can easily make out that water nucleophilic attack is actually the pathway for oxygen-oxygen bond formation. because the reaction is first order with respect to our catalyst concentration. So we can rule out easily that it is not an ITO mechanism, because had it been an ITO mechanism, the reaction should be second order with respect to catalyst concentration. So just by doing a simple kinetic study, we can make out that which pathway actually we are forming an oxygen-oxygen bond. So based on our kinetic study, based on our electrochemical study, and also some spectroscopic study, which I have not shown here, so we propose this particular reaction cycle. So this is our starting ruthenium-2. So it oxidizes by 1 electron and forms a ruthenium-3 aqua. Then it undergoes couple of proton-coupled electron transfer as well as electron transfer to form a high-valent formally ruthenium-6 oxo species. And once it reaches at ruthenium-6 oxo species, then a water nucleophilic attack takes place, and it forms a putative ruthenium-4 hydroperoxide intermediate, which then undergo further oxidation and then ultimately evolve oxygen. So, although we understood that how our in this particular case how the oxygen-oxygen bond formation is taking place, but now we want to know, although these two complexes having a similar type of coordination environment, however, two aqua complex is showing much better activity compared to one aqua complex. So to investigate that, we perform a very simple experiment. What we did, we oxidize our ruthenium-2 complex by 2 equivalent to form a ruthenium-4 oxo species, and then we perform an 1H NMR analysis. So this is our starting 1H NMR species, 1H NMR for the ruthenium-2 species. As you know, it is a D6 S0 diamagnetic, so it is showing a very nice clean sharp NMR spectrum from 0 to 10 ppm. But when we oxidize it by 2 electron, is retached to ruthenium-4. Now it's a D4, and if you look at the d electron configuration in octahedral, then D4 has a 2 unpaired electrons. It's a 2 electron paramagnetic. So we are seeing a paramagnetic effect in our 1H NMR spectrum. It's become line broadening is happening due to the paramagnetic effect. So this is nothing surprise, because in this case it is 2 electron paramagnetic. So we would expect there should be some paramagnetic effect in your 1H NMR spectrum. But on the contrary, when we perform the similar experiment for our one aqua complex, with our surprise, what we see at ruthenium-4 state also, it is showing a very nice NMR spectrum from 0 to 10 ppm range. So which indicates that for this complex at ruthenium-4 state, the complex is diamagnetic in nature. Now, how you are going to explain this particular phenomena? So if you look at now the d orbital splitting diagram, so in octahedral, you have this T2G and EG d orbital splitting, and this is the at low spin state, this is the electronic configurations. So at octahedral geometry, your ruthenium-4 state is 2 electron paramagnetic in nature. But if it expands its coordination number from coordination number 6 to coordination number 7, and if it adopts a pentagonal bipyramidal geometry, then your T2G and EG level will further split, and then we will have this type of d orbital splitting diagram where all the electrons can be paired up. So that particular case, your system will become diamagnetic in nature. So what we believe that for this particular case, for this particular N4PY system, so actually when we oxidize it at higher valence state, the molecule expands its coordination number from 6 to 7, and then it adopts a pentagonal bipyramidal geometry. But for the two aqua complex, it remains its octahedral geometry even at higher valence state. So we believe that makes the difference in their reactivity, because when you have an extra ligation at higher valence state, now my ruthenium center is getting some extra electron density from this extra ligated water molecule. So we can expect that the electrophilic character of this oxo moiety will be reduced. But in this case, it remains its octahedral geometry even at higher valence state. So its electrophilic nature for this Ru oxo moiety will be much higher compared to this particular case. So what we believe that in this case, water nucleophilic attack cannot take place effectively, but it is happening in much better way for when it maintains its octahedral geometry even at high valence state. So although our complex, this particular complex is capable of doing oxidation of water, but we have get only a handful number of turnover numbers. We have achieved only turnover number of 6. This is not at all good in 2020 because there are lot of catalyst has been reported in literature which has a very high turnover number. So with respect to that, this particular complex, although it is performing the oxidation of water, but we are not getting a very high turnover number. So what we believe that beside this doing the oxidation of water, a side reaction is going on, which actually derailing the catalytic activity. Now to understand what could be the deactivated product or what could be the degraded product, so we performed a mass spectroscopic study of our post catalysis solution. So what we observed that after oxidation, this benzyl group actually detached from our ligand system, from our complex, and it forms this type of complex where water also get replaced by your counter anion present in the solution. So as it's lost its water coordination, it's lost its activity. And this benzyl group gets oxidized to form a carboxylic benzoic acid which we have detected by GC analysis, and this can undergo further decarboxylation under oxidizing condition to form a trace quantity of CO2, which we also have detected in our GC analysis. So from this particular system, what we have learned that if we want to make a very high turnover number, then our catalyst design should be such that that organic ligand should not possess a very easily oxidizable moiety. It should be oxidatively robust in nature. So keeping this lesson in mind, now we develop a second generation of catalyst in our lab. So we use a trianionic tridentate ligand to make a ruthenium complex. So this is the synthetic procedure. This trianionic ligand, it's a meridional ligand. It occupies the meridional positions of the octahedral geometry, and the other 3 positions is occupied by the 3 other picolyl groups. So why we choose this trianionic ligand? Because we know so this type of ligands will be having a very strong sigma donating effect, and that can actually help me to stabilize the ruthenium at very higher oxidation state. So that was our purpose. Not only that, this particular ligand also can participate in redox reaction. So this is a redox non-innocent ligand. So in principle, it can involved in 2 electron oxidation process. So first electron removal can generate a radical, second electron removal can generate a positive ion. So as this ligand is redox non-innocent in nature, and our metal is also redox non-innocent in character, so we can expect a metal ligand cooperativity in acquiring the oxidizing equivalent in a single molecule. And this is very important for developing a catalyst for performing an oxidative transformation. Your catalyst should be capable to acquire oxidizing equivalent within itself. So this particular complex we have also characterized by single crystal X-ray crystallography. So here I want to emphasize its one structural parameters. So if you look at the bond distance between ruthenium and equatorial nitrogen and the ruthenium, this particular nitrogen bond distance, the equatorial picolyl group is much higher compared to this particular bond distance. So this is actually is due to the trans-levelizing effect observed of this anionic nitrogen group. Now, as I told you, that to perform an oxidative transformation reaction, you should have in water ligations. So although apparently this particular metal complex does not have any water ligations, but due to this trans-levelizing effect, what we could expect that this particular equatorial position will be very much labile in nature. And once we dissolve our complex in water, we expect that water can replace this equatorial picoline group, then we can have a water ligation. So in principle, we can displace two positions of picoline groups to form two types of isomers. So in this case, axial displacement may occur. It can form a axial isomer, axial ago-ligated isomer, and from it can also displace this equatorial position to form this equatorial isomer. But from the translabelizing, due to the translabelizing effect, what we exclusively get this equatorial isomer. We don't get any axial isomer. So from DFT study also, we have calculated the relative energies. What we found that this equatorial isomer is 5.87 kilocalorie per mole, is more stable compared to this axial isomer. So once we dissolve our compound in water, so axial picoline group gets displaced by water, and then we generate our real catalyst, aqua-ligated metal complex in situ. So this is the electrochemical behavior of this metal complex in water. So as you can see, it shows a couple of redox responses. So for time being, let's assume that all the electrons we are removing from the metal centers, although it may not be true, because as I told you that our ligand is also redox non-innocent in nature. But for the time being, let's say that let's assume that all electrons we are removing from the metal center, just to have an count how much formal oxidation state of ruthenium we can achieve for this particular system. So our first wave we assign is due to the ruthenium 2 to ruthenium 3. Second wave is from ruthenium 3 to ruthenium 4, and the third wave from our bulk electrolysis, what we observe that this particular step, actually we are not removing one electron, but we are removing two electrons. So that means we are generating a from ruthenium 4 to ruthenium 6 species at this potential. Now once it reaches to ruthenium 7 formally, it's then started doing triggering oxidation of water. So we have performed a detailed Pourbaix diagram also. As you can see that it involves, shows a couple of proton couple electron transfer reaction. That also indicates that you have a water ligation in your metal complex. So all the way when you are oxidizing, when you are applying or increasing the potential, all the way we can generate a formally high valent ruthenium 7 oxo species. So ruthenium 7 oxo species formally, but it may not be true because our ligand is also redox non-innocent in nature. So in some steps, so ligand center also may undergo oxidation. So now to know that proper electronic structure of these various oxidized species formed during the oxidation process, so we performed DFT calculation as well as we performed the EPR spectroscopic study. So this is our starting point. So at our starting point, our ruthenium is +3 oxidation state that we proved from our EPR spectrum. Because in EPR spectrum, we are getting a rhombic type of EPR signal, which is very characteristic for low spin ruthenium 3 species. So at our resting state, our ruthenium is in +3 oxidation state. So now once we oxidize by one electron, so we formally generate a ruthenium 4 species, but from DFT calculation, it shows that actually we are not oxidizing the metal center. Rather we are oxidizing the ligand center. It is forming now a radical. Now this ruthenium 3 in low spin D5 system, so it has a 1 unpaired electron, and then your radical has an 1 unpaired electron, and that 2 unpaired electron, they interact ferromagnetically to form a S equal to 1 ground state. Now once we remove a 2 electron, 2 proton simultaneously, we are reaching a ruthenium 5 oxo species. Although a formal oxidation state is ruthenium 6, but actual electronic structure is a ruthenium 5 oxo species, and you have a radical. Now in a ruthenium 5 at low spin state, it also has a 1 unpaired electron, and your ligand is also an unpaired electron. So they also interact ferromagnetically and forming a S equal to 1 ground state. As these two species has a S equal to 1 ground state, so they are showing a very similar type of EPR spectrum. So what we observed a half field signal here for these two particular species, which is this half field signal is actually very characteristic when you have an S equal to 1 ground state. So from the DFT calculation, so whatever we have predicted from DFT calculation, their electronic structure, this actually is supported by our EPR study. Now in last step, once we remove our last electron, so that means formally we are generating a ruthenium 7 species, but in actual electronic structure, that we are actually oxidizing one more of our ligand is forming a plus charge. So this is the proper electronic structure of this particular ruthenium 7 species. Now from this is the spin density, molecular spin density map of this particular species. So from where we can see that this particular oxygen atom also contains a substantial radical-like character. So that means this ruthenium 5 oxo, it has a radical-like character. So then we can expect a radical coupling pathway for making the oxygen-oxygen bond. So we perform this kinetics analysis also as before. So we use steric ammonium nitrates in this case also for as a sacrificial oxidant, and we study oxygen evolution kinetics by varying the concentration of complex. So like before, here also we are seeing that rate of reaction is linearly dependent on complex concentration. What does it mean? That it means that reaction that only one complex molecule is involved in rate determining step. But when we vary the cerium concentration, unlike before, now we are getting a saturation type of kinetic trace. So this type of saturation trace we can actually model using a Michaelis-Menten kinetic model, where enzyme and substrate, they form an enzyme substrate adduct and then remains in an equilibrium before a rate determining product release step. So using this model, what we proposed for oxygen-oxygen bond formation, so as I told you before, that this cerium 4 hydroxide, it also contains a radical-like character, and from the molecular spin density calculation, we observe this particular species also have a radical-like character. So what we expect here, that a oxygen-oxygen bond formation takes place by heterometallic pathway and forms this ES type of adduct, which remains in an equilibrium before a rate determining oxygen evolution step. And from this kinetic stress and also some by other fitting, we actually can calculate the rate constant value, the equilibrium constant value, and all other rate constant value, which I am not showing here. Now, in this case, as you can observe that that steric ammonium nitrate is not only acting as an sacrificial oxidant, but it also helping in making an oxygen-oxygen bond. Now, the radical-like character of this cerium is indeed needed for this particular case to make an oxygen-oxygen bond. So we performed is we now changed our oxidant from cerium 4 to sodium periodate. Now, you know that sodium periodate is an oxo-transferring agent. It does not have a radical-like character. So now if you compare the oxygen evolution rate, but using CAN and sodium periodate as an oxidant, you see for periodate, we see a much lower activity compared to cerium. So that implies that indeed a radical-like character is needed to make an oxygen-oxygen bond for this particular case. So this is the overall summary of this molecule, how it is promoting the oxidation of water. So once more, I am summarizing it here. So this is our starting point. So once we dissolve this particular compound in water, so equatorial picoline is replaced by water, and then I have a water coordinated real catalyst. Now it then oxidizes by 1 electron, and from the DFT calculation as well as from the EPR study, we conclude that actually we are not oxidizing the ruthenium center, rather we are oxidizing ligand center, and then it's from this particular species, and then after 2 electron 2 proton PCT reaction, it forms this species, and ultimately it generates this particular species where we see a radical-like character on oxo moiety. And then a heterometallic coupling takes place between cerium and this particular species, and from this particular intermediate, which then after that a decoordination of cerium happens, and then you form a hydroperoxo type of intermediate 1, and then further it that OH bond breaking takes place, and then it ultimately releases oxygen. So from we also, when we replaced this hydrogen with deuterium, we observed a KIE effect of 3.1. So we conclude that in this overall catalytic cycle, this OH bond breaking is the rate determining step. So now I move to a third example of catalyst. So this is a part of my postdoctoral work. So here also we developed a mononuclear ruthenium complex by having a pyridyl as well as carboxylate type of donor groups. And this with this particular complex, we achieved the highest turnover numbers and highest turnover frequency. This particular complex actually catalyzed the reaction is a very high rate. We achieved it more than 300 turnovers per second, which is very much comparable with our natural photosystem too. And because of that, up to 2012, for a homogeneous system, this was the uh, this was the highest performing catalyst. And because of that, we could publish this particular work in Nature Chemistry. So here one thing I want to emphasize here about structural parameters. So the oxygen ruthenium, the equatorial oxygen ruthenium oxygen bond angle, you see it's a 123. It's a quite expanded compared to your normal 90-degree octahedral angle. So that means that from equatorial position, it has a lot of space so that our compound can expand its coordination number from coordination number 6 to 7. So this particular angle allows the complex to expand the coordination number from 6 to 7. So indeed, we have observed, we have isolated uh, a we have isolated a 7 coordinated ruthenium 4 species, and we have characterized its crystal structure also. So once we oxidize our complex from ruthenium 2 to higher valence state, so an extra ligation takes place from the equatorial site. So this is the electrochemical behavior, as you can see. So this complex also show a couple of proton couple electron transfer reaction, and then ultimately it reaches to a ruthenium 5 oxo species. Now from the DFT calculation, we found that this is not the ruthenium 5 oxo, rather it is a ruthenium 4 oxyl intermediate. So this oxo has a very high radical-like character. So once you have a radical-like character, so from the kinetic analysis, we have concluded that the oxygen-oxygen bond formation is taking place by a bimolecular pathway, by an I2M mechanistic pathway, because the reaction is second order with respect to your catalyst concentration. And here the pi-pi interactions of this axial moiety it also helps to bring together two molecular unit and form an oxygen-oxygen bond in a bimolecular way. So here pi-pi stacking favors the oxygen-oxygen bond formation, and because of that, its turnover frequency is very high. So far I have discussed about only the mononuclear compounds. So let me talk about some multinuclear compounds which we can use as a water oxidation catalyst. So for this purpose, we have synthesized this type of heterotrinuclear compound. So it's a ruthenium M and ruthenium 2. So our M is cobalt and manganese. So we have synthesized two types of metal complexes with acetate. We have isolated this particular trinuclear compounds where we have acetate ligations. Why we made an acetate? Because we know the acetate is a very labile in nature. So once we dissolve this complex in water, acetate can be easily replaced by water. Then we will have a water coordinated complex in situ generated, which can act as a real catalyst. So our purpose of making this particular trinuclear compound to see is there any cooperativity effect among the metal centers to promote the oxidation of water. So to know that whether our complexes, all the metal centers are in communication or whether they are in a talking term or not. So to know that, we perform this electrochemical study. As you can see, it shows a very beautiful, well-defined CV responses. So because we are getting 3 oxidative redox responses because we have a 3 metal centers. So that indicates that our metal centers are communicating with each other. And at various oxidation state, we have performed this EPR study. Let me explain this particular EPR spectrum. So our starting point, we have a ruthenium 2, cobalt 2, and ruthenium 2. So as you know, the ruthenium 2 centers, it's a low spin D6, that means it's a diamagnetic in nature, Once you do not have any unpaired electron, that means ruthenium-2 center will not have any contribution towards the EPR spectroscopy. But cobalt-2 in high spin state, it has a 3 unpaired electrons. So we are seeing an EPR spectrum typical for the high spin cobalt-2 for this particular species at resting state. Now, when you oxidize it by 1 electron by applying a proper potential, we are generating a ruthenium-2, cobalt-3, and ruthenium-2 intermediate species. So as you know, in all three metal centers, it's a D6 system, low spin. So there is no unpaired electron. So it's become EPR silent. Now, when we oxidize by 2 electrons, the terminal to ruthenium 3,3, so we are generating a ruthenium-3, cobalt-3, and ruthenium-3 species. Now, you know that cobalt-3 is a D6, there is no unpaired electron, so it does not have any contribution in EPR, but ruthenium-3 at low spin state, it has now 1 unpaired electron. Now, apparently it looks like that these two ruthenium are very far to each other, but actually they can communicate through the middle cobalt center, and actually these two unpaired electron or two terminal ruthenium, they interact ferromagnetically and form an S equal to 1 ground state, because we see an half field signal here. So from this EPR spectrum, we can confirm that our metal centers are in talking term. So they are communicating with each other. That means we can expect some sorts of cooperativity effect when it promotes that oxidation of water. Now, to check whether this particular complex is capable of doing oxidation of water or not, we first check its electrochemical behavior. As you can see, it shows a very high catalytic current, which indicates that it can trigger the oxidation of water. And not only that, we also could detect the reduction of oxygen to superoxide, which is generated during the water oxidation at this particular potential. So from this particular study, we now confident that we make sure that this complex is performing, fulfilling our purpose, that is, it is doing that oxidation of water to make oxygen. So we performed, now in this case, we have studied a photosensitive photochemical oxidation study. So for that, we use ruthenium tris-bipyridine as a photosensitizer. So when you flash with light, so this ruthenium-2, it goes to the excited state, and at excited state, its reduction potential decreases enormously, and then it can be easily oxidized by sodium persulfate. This acts as a sacrificial electron acceptor, and then you can generate easily a ruthenium-3 species. So this ruthenium-3 then oxidize our catalyst and to go to higher oxidation state, and then our catalyst is doing the oxidation of water to make oxygen. So this is the oxygen evolution time trace of various catalysts. So our ruthenium cobalt, that analog is showing much better activity compared to manganese analog and also compared to mononuclear ruthenium. Now, to know how the oxygen-oxygen bond formation is taking place for this particular case, so we performed a leveling experiment using oxone as a sacrificial oxidant. So from this leveling experiment, what we have concluded that actually water nucleophilic pathway is the pathway for oxygen-oxygen bond formation, and that is taking place at the middle cobalt centers. And the terminal ruthenium centers, the water or oxo is coordinated at the terminal ruthenium center. They are actually helping in hydrogen bonding interactions to stabilize this particular intermediate. So by that way, they are all three metal atom in a cooperative manner is triggering the oxidation of water. So in summary, so I hope I could show you various types of catalyst and various types of pathway for making the oxygen-oxygen bond to promote these particular reactions. So one thing you have to keep in mind, if you want to increase your turnover number, so your catalyst design should be such that it should not have a easily oxidizable organic group. And also we could show that redox non-innocent ligand, nowadays this type of ligand has become a very hot topic in research. So redox non-innocent ligand also you can utilize in acquiring the oxidizing equivalent in your catalyst. And with this particular example, we could show that a heterometallic O bond formation is taking place via radical coupling pathway. And sometimes also the secondary interactions like pi-pi stacking interaction is also helps in making the oxygen-oxygen bond. And in a multinuclear complex, the cooperativity effect among the metal center also helps to promote the oxidation of water. So this is the with acknowledgement. So this is my group. I have very small group with 4 PhD students. So the first two parts which I did from the IIT Kharagpur, that particular result is done by on MS Kundu and a postdoctoral student, Dr. Suman Kumar De. And in that project, two master students, Chiranjit Datta and Srimoy Khan, they also contributed a lot. I collaborate with my departmental colleague, Professor Anu Payappan, for the DFT calculations. I especially thanks to Professor Anthony Lovett. He was my mentor during my postdoctoral study. And these are the money source, DST Inspire Faculty Award, IIT Kharagpur Startup Research Grant, and SCRB India. And I also thank Department of Chemistry for giving these infrastructural facilities. With this, I thank you all for your kind attention.
Yeah. Well done, Doctor. You have shared your research findings and other things. Okay, it is a very good, very informative and innovative things. Okay, when we think about your research, we will elaborate all the things. Okay, for human benefits. Okay, Yamuna,
Sir,
any question is there in this
Yes.
chat?
yes, sir.
Yeah, please read it.
Okay, sir.
Yeah, so I will be happy to answer, or at least I will try.
Sir, one of our participants has asked this question, sir. In the slide 12, for ruthenium 3OH2 compound, why does the reduction potential decreases with increase in pH?
In slide 12,
Yes, sir.
okay, why the reduction potential decreases with increasing pH? Because
Yes, sir.
if you follow the Nernst equation, so the Nernst equation, it says that your E is equal to E0 minus 0.059 pH. Okay, so when a proton transfer is involved, then that is your Nernst equation. E is equal to, that in formal potential, E is equal to E0. E0 is your standard potential minus 0.059 pH. Okay, so that means when you increase the pH, you are actually decreasing its potential. That means when you increase the pH, you are making easy to oxidize your species.
Thank you, sir.
It's become easier to oxidize your species.
Yamuna,
Sir?
any more question is there?
Hello, hello, Professor.
Yes.
Thank you for very nice talk. I have one question regarding this EPR spectra for your non-innocent ligand and also your multinuclear ligand, multinuclear complex. You show that use S equal to 1 in the EPR spectra. I mean, how
Yeah,
it's
S
possible?
equal to 1 generally become EPR silent because of the Kramer's degeneracy. But sometimes we may observe that this MS equal to 2, this is actually a spin forbidden transition. Okay, so that's why sometimes we may observe, we may not observe. So as it is a spin forbidden, so intensity sometimes become very low, or it becomes very broad that we don't see. But in our case, we see. But once we see it, in some cases, there is an evidence we can see it. Once you see it, then definitely that confirms that you have a S equal to 1 ground state.
And this this one that you showed the circle, that is the characteristic for S equal to 1.
Yeah, because this circled one, this is a spin forbidden transition. Your spin allowed transition is delta ms equal to 1. That will be your allowed transition. Okay, so in that cases, you will see a very sharp signal. But sometimes we also observe some forbidden transition. So for s equal to 1, this small intense peak what we are seeing, that is delta ms equal to 2. Okay, and that is a spin forbidden. That is a forbidden transition. But once we of it only happens when you have a S equal to 1 ground state.
Okay. And
Because when you have a S equal to the ground state, then you can have a splitting like ms equal to 0, ms equal to plus minus 1. And then when you apply magnetic field, so your plus minus 1, that energy will get splitted. And then you are going to see some spin forbidden transition from -1 to +1. Okay, so if you just study the EPR part, so I think you can easily understand.
okay.
Yamuna, any more question is there?
Hello,
Yeah.
Mandal sir.
Yes.
Sir, I want to ask one thing. Sir, if you, yeah, sorry, you are mentioned in slide 8 and 9, the oxidation of water to produce the oxygen and hydrogen gases molecule. Sir,
Yes.
if excess amount of hydrogen and gases molecule are produced, then if it may cause of the imbalance of the climate also. I mean to say, sir, climate exchange, then it is possible to control this oxidation through the any reverse oxidation process, as it is very highly endothermic also. Can any kind of reverse oxidation for this process controlation is possible, sir?
I mean, you mean to say that if you produce a lot of hydrogen, that might cause some abnormal
No,
things?
no, sir. Hydrogen may be used in hydrogen fuel cell,
Yeah.
but the amount of oxygen also left it out simultaneously.
But that
Sir,
oxygen is not that harmful, no? Because and oxygen, the oxidizing, because the oxidation with oxygen sometimes is a very kinetically slow process. Because you know that we are living in an environment of oxygen, but why then oxygen is not harming us? Why our skin does not get burned by the oxygen? Because we are living in an oxidizing environment.
sir,
Because this is a pin selection rule forbidden, because our skin is a it's a organic molecule, so it's a diamagnetic, I would say, and oxygen it's a triplet. So triplet, singlet, that does not interact. But had it been in a singlet oxygen, then we are in danger. Okay, so this is, but
sir,
fortunately oxygen is triplet state, at ground state is triplet. So that's what, that is actually saving us to getting burnt out in an oxidizing environment, and you can easily survive. To activate the oxygen, you need a metal catalyst. That is actually our various enzymes are doing in our body.
yes, sir. Sir, if oxygen-free radical can also be produced the ozone molecule, that can be helpful. But
Yeah,
there is something problem as corresponding to the carbon dioxide is an also an our important element, but it's produced the global warming. Simultaneously, if higher amount of oxygen is produced, then it may causes on the our climate
no,
field of
you see,
percentage.
if about the what is the scheme is showing, so you are not actually producing a lot of oxygen, because there
Yes,
will be a
yes.
balance. Because why? You see, if you combine this this particular fuel cell as well as the artificial photosynthesis device, so what we are doing, from water molecule you are making hydrogen and oxygen, and then you are utilizing those things and you are making water. So that means there will be some balance, what you produce, how much amount of oxygen you are producing, and how much amount of water is getting produced. So I don't think so with this process you are going to increase the level of oxygen in atmosphere.
Okay, sir. Thank you for your nice talk, and thank
Thank
you,
you.
sir.
Yamuna, any more question in chat box?
Yes, sir. We have another one question.
Yeah.
Can ruthenium be distinguished?
I cannot hear properly.
Sir, now it
Yeah.
is okay, sir.
You can unmute.
Yeah.
Yes, sir. I'll read it again, sir. Can ruthenium 4 be distinguished from ruthenium-3 and BOT by EPR as both are is equal to
Well,
one
your
species?
voice is sometimes getting up and down, so I cannot hear properly. I think
Yamuna, you can
you
repeat the
repeat?
question.
Can
Yeah,
you
Yes,
your
repeat?
sir.
voice is problem.
Yes,
Yeah.
sir.
I will read out
Okay,
read out
okay.
the question. So the question is that it is raised by Annex Jose from Stanford.
Oh,
Can ruthenium 4 be distinguished from ruthenium 3 N dot by EPR as both are S is equal to one species?
ruthenium 3. Can you repeat once more?
Hello.
Yeah, ruthenium 3. Ruthenium 3, I suppose it's a S equal to half.
You have to compare with ruthenium 4, doctor. Yeah.
Yeah, ruthenium 4, I think it's a S equal to 1, and ruthenium 3 is S equal to half.
Oh.
Ruthenium is S equal to half, but the actual question is ruthenium 3 N dot, the radical, nitrogen radical. How to
Ruthenium.
he is asking ruthenium.
Okay. So formally that is a ruthenium 4, but in our cases it is happening. You have a ruthenium 3 1 unpaired, and then 1 dot. So they are ferromagnetically coupled, and that's why we are getting S equal to 1 ground state. That is actually predicted. That is actually predicted by DFT. So we took help from the DFT. So
Yeah.
DFT suggests we have calculated in various spin state energy, and we found that in triplet state that particular molecule is the ground state.
So how to distinguish these two S is equal to one species by EPR? That is the question, actual question.
S equal to 1 by EPR means if you get an EPR half field synchrotron, that tells you have a S equal to 1. Now how that S equal to 1 is coming, whether two spin like in case of nickel also, nickel, that is also an S equal to 1 system. But in our case, two unpaired electron at two domain, they are interacting, and that is making S equal to 1 system. Okay. So from EPR, I cannot say that how this S equal to 1 is forming. It only can suggest that you have an S equal to 1 ground state, right? So as I told that from DFT calculation, we have calculated at various spin state their relative energies, and we found at triplet state that particular molecule is the ground state. So we actually our EPR DFT study is complement with EPR.
Sir, we have another one question. Can you comment on the major limit?
Can you comment on the major?
No, sir. Major
Can
limit.
you comment on the major limitation factor in water oxidation?
Okay, as I told my introduction that I am mainly talking about the molecular system. So the major limitation of molecular system is the stability of your ligand framework. So nowadays we have achieved a lot of turnover number, lot of turnover frequency, but it's not very stable for a long time, so that we can actually use it to develop a technology. So from the coordination compounds point of view, the major limitation what I felt is that our ligand system is not that much stable enough, oxidatively robust enough, so that we can use it for a very long period of time.
Any more clarification, Emna?
No, sir.
Okay, sir. Professor Mandal, very good informative session. And I have two clarification, Professor.
Yes.
Yeah.
Sure.
One is in your presentation, you have mentioned delta G naught, I mean standard free energy plus 113.38 kilocalories per mole. Okay.
Yeah.
Usually, normally photosynthetic route, usually negative free energy, isn't it?
No, this is from the water to oxygen, that particular
Water
energy.
to oxygen. Okay.
Water to oxygen.
Yeah. And one more clarification, Professor. Okay. Usually, it is a general question only. Okay. You have compared with the artificial and natural photosynthetic route. Okay. So when we see that light energy, when we fall into our leaf, I mean chlorophyll, okay, it's converting into reacted with the carbon dioxide and water, it's converting into starch or carbohydrate.
Yes.
Is it right?
Yes.
Yeah.
That happens in photosystem 1.
Photosystem only.
Yeah,
where we are lacking in that artificial photosynthesis, because we are not generating starch and
no. You see, in natural photosystem 2, how we store solar energy? We store solar energy in the form of a chemical bond energy in our carbohydrate.
Yeah, where we are lacking, Professor, that
Sorry?
Because it is a challenging task now. When we make it that one, we will avoid photosynthesis also. In artificial, when we make it that short synthesis.
No, that's what I'm telling. In natural photosystem 2, natural photosynthesis, how we store, where we store the solar energy. So we
Okay.
store solar energy in the form of a chemical bond energy in your carbohydrate. Because you saw that photosynthesis is a two parts. One is photosystem 2, one is photosystem 1.
Okay.
So in natural system, in photosystem 2, it oxidize water and it take out the electron and proton from the water. And
Okay.
with that electron and proton, it reduce your carbon dioxide, and that means into your carbon dioxide, that particular electron and proton which you are taking out from the water, that is stored in the form of a carbohydrate.
Okay.
Okay, so that is how we are storing solar energy in the form of our food. And when you take that food, we are actually taking that energy back,
Okay.
right? So, but in this case, we are not storing that electron into carbon dioxide. We are storing that electron in hydrogen molecule.
Okay.
Okay, so for any compound which you can use as a fuel, that should be reducing in nature, so that we can get some electron out of that. So any reducing agent which can donate the electron, which can be a source of electron, that we can use as a fuel. So that's why hydrogen, we are storing the electron in hydrogen. That's why hydrogen can act as a fuel.
Okay.
Doctor, one more question.
Yeah.
One more question. So wait a minute, I will unmute the participant. He will ask the question. Wait a minute, doctor.
Yeah, sure.
The student will ask the question now. Prabhakaran, you can ask the question. Now you are unmuted.
Not at that unmute. Yeah.
So one more question raised in the chat box is that the major limiting factor in water oxidation. Can you comment on the major limiting factor in
major limitation from the point of view of energetics? Okay, this oxidation part of water. You see, if you this oxidation part of water that I have already told you in my introduction. So you see, so this developing a photosynthesizer, that is one domain of research. Developing a water oxidation catalyst, that is one domain of research where I am interested. Okay, and developing a proton reduction catalyst, that is also one domain of research. And when you come to the fuel cell technology, the reduction of oxygen is taking place. So reduction of oxygen, that is also one domain of research. So all research outcomes should come together and they should work in harmony, so that then only we can develop, we can dream of that we can drive our car with water and sunlight.
Oh. So, Prabhakaran, you can raise your question. I think somebody asked, but he is not. I think he is not available. Suresh sir, you can now give the.
Yeah. And Professor Mandal, thanks for your very good session. Okay. On behalf of FCS 2020, we are thankful to you for your good time with us. Okay.
Thank you very much. So, so once more, I congratulate all of the organizing committee members to organize such a successful webinar. It's a the topic you have chosen, it's a relevant to my research interest. I thoroughly enjoyed yesterday's two lecture, and I am also looking forward to listen other lectures also. Thank you very much.
Thank you, doctor. Thank you so much for your nice presentation. Yeah. Thank you, Suresh sir, for sharing the session. I thank one and all for your active participation. The afternoon session will start by 2 o'clock sharply, and all are asked to log in before 15 minutes before into Zoom as well as YouTube link. Thank you so much. The feedback links are available in the chat box of YouTube as well as in the Zoom. So you all are asked to fill the feedback link. Thank you so much.
So can I leave now?
Yeah, sure, you can.
Okay.
Yeah. Thank you, doctor. Yeah.